Rahim. My name is Wesan and I have to give a presentation under two topics, neural crest cells and sapelogenesis. So first of all, we look into the neural crest cells. Neural crest cell. At the time of formation of the neural plate, some cells at the junction of the neural plate and surface ectoderm become specialized. As you can see in the first diagram, there is epidermis or the surface ectoderm. They converge and give rise to the neural fold. At the formation of neural fold, the neural plate, there are some cells that specialize. As you can see in the last diagram, the green cells, the tiny cells, they are the neural crest cells. With the separation of the neural tube from the surface ectoderm, neural crest cells lie along dorsolateral sides of the neural tube, become free and migrate throughout the body, divide into dorsal mass and ventral mass. They divide, neural crest cells divide into dorsal mass derivatives and ventral mass derivatives. So we now look into the dorsal mass derivatives and ventral mass derivatives. That what is a dorsal mass derivative and ventral mass derivatives. First of all, we discuss the dorsal mass derivatives. Neuroblast. Neuroblast give rise to pseudo-unipolar neurons of dorsal root ganglia of spinal nerves. They also give rise to the neurons of the sensory ganglia of 5th, 7th, 8th, 9th and 10th cranial nerves. Spongioblasts. Spongioblasts form capsular or satellite cells of all sensory ganglia. They also form Schwann cells, which form the neurolemma and myelin sheaths of our peripheral nerves. Pluripotent cells. Pluripotent cells form the mesenchyme of dental papilla, odontoblasts and dentine. They also form melanoblasts, which is the pigment cells of skin, cartilage cells of branchial arches, and leptomeninges. Leptomeninges form the pia mater and arachnoid mater. Ventral mass derivatives. Sympathetoblast and chromaffin cells are the ventral mass derivatives. Sympathetoblasts are small cells while chromaffin cells are large cells. Sympathetoblasts give rise to the neurons of sympathetic ganglia, neurons of peripheral parasympathetic ganglia of cranial nerves. Chromaffin cells. Chromaffin cells give rise to the adrenal medulla, para-aortic body, argintophin cells, and enterochromaffin cells. These are the different contribution of cranial nerve crest to the craniofacial tissues. Now we look into the sapelogenesis. Sapelogenesis means formation of head. Neural tube expansion. As we discuss neural tube formation, neural tube sinks beneath surface ectoderm. The anterior portion of the neural tube expands and gives rise to forebrain, midbrain, and hindbrain. Hindbrain develops a series of 8 bulges, which are rhombomeres, or which are called rhombomeres. Paraxial mesoterm partially segments rostrally to form somatomeres and segments caudally to form somites. Rostrally and caudally are different positions like anterior and posterior, the upper end and the lower end. So the upper end are rostrally side give rise to somatomeres, and caudally side, which is the lower side, are the posterior side, which give rise to somites. Occipital somite is the first somite form. Rhombomeres. As we discuss how the rhombomere form, so now we discuss the contribution and formation and how they help in the sapelogenesis. Neuroglial cells from 2 rhombomeres migrate as 2 streams to supply additional embryonic connective tissues needed for craniofacial development. The first 2 rhombomeres, they migrate and give additional embryonic support to the craniofacial development. The first rhombomere provides ectomesenchyme associated with the face. The second stream which arises from the second rhombomere contribute to the formation of jaws. Neurocrest cells from rhombomere 3 and beyond migrate into arches that will give rise to pharyngeal structures. Homeobox gene. The general definition of homeobox gene is that homeobox genes are a large group of similar genes that direct the formation of many body structures during early embryonic development. So now we look into the homeobox gene, that which homeobox genes help in the formation of head, or which help in cephalogenesis. So different sets of homeobox gene are adapted for the development of cephalic structure: orthodentical homeobox 2, muscle segment homeobox, the distal-less homeobox, the bar H-like homeobox. They are different homeobox genes which help in the cephalogenesis. Homeobox transcription gene factor are not expressed anterior to rhombomer 3. Common signaling pathways. Some neurocrests require instruction for their local microenvironment. These required instruction involved common signaling pathways like hedgehog SHH, sonic hedgehog, fibroblast growth factor, and bone morphogenic proteins BMP. Patterning of head and face. Enzymes that modify chromatin's architecture also participate in craniofacial patterning. So different enzymes are involved in the formation of craniofacial patterning. Species-specific patterning of the head and face, especially the shape and size of beak and muzzle, depend on canonical WNT signaling pathways. These pathways are an upstream modulator of critical effector molecules such as FGF8, BMP2, and SHH present in the frontonasal ectodermal zone center. Very different shape encountered in the nature are due to the variation in the organization, relative size, and position of FEZ, which is the frontonasal ectodermal zone or center, along with the molecules like calmodulin. The calmodulin and FEZ center, their variation and different organizations and their size and position gave rise to different type of shapes of the head, or different type of the heads that we encounter in this universe. Thank you, and if you have any question you can ask.